What's up, science fam? So you guys should already know by now that cells come in many sizes and shapes. But how exactly do cells survive? How are they the fundamental unit of life if most of them are so small? So this week our topic will be about cellular structures and how cells can live on their own. So let's get right into it. So how can cells survive when they're so small? Again, this is due to the cellular structures, or also known as the organelles. So that's a bit of a confusing term. We're going to clarify that later on. So in this set of videos, we're going to differentiate the types of cells based on their characteristics and their morphology. We're going to describe the structures that make them up, and we're going to relate these structures to the actual functions of these structures and how they help cells, different types of cells, survive. So the first thing we should ask is, do all cells have the same structures? That's part of some of your activities. So the short answer is that no, different cells have different structures. And it depends on what functions your cell needs to do, what structures it would need for it to perform well and to thrive in its environment. So we're going to focus on three types of cells: the plant cell, the animal cell, and the bacteria cell. We're going to go through the structures of each one. So we're going to go through this in this format. So what holds the blueprints and the instructions, what kind of molecule directs the fate of the cell? That's basically the DNA, or the deoxyribonucleic acid. It's the genetic material. And all cells need to have DNA. So where does that get stored in the cell? So it depends on the type of cell. For eukaryotes, it's kept in the nucleus. It keeps the DNA locked up safe inside the nucleus. In fact, that's where you get the name eukaryote, meaning they have a true nucleus. So these are some of the parts of the nucleus which we'll describe in a bit. The nucleoid region, on the other hand, is a blob of floating DNA in your prokaryote. So you can see here the cell wall, some ribosomes. The nucleoid just looks like bleh, just the DNA floating around. So describing the different parts in the nucleus, we have the nucleolus, a small ball inside the nucleus that assembles ribosomes. The outer covering of the nucleus is the nuclear envelope. So it's a double membrane system, meaning it has two layers, two skins. The nuclear pores are the only way in and out of the nucleus and through the double membrane, which is the nuclear envelope. And the chromosomes are basically the coiled up versions of your DNA molecules. In your prokaryotes, on the other hand, the nucleoid region is basically the bacterial chromosome. It's a single big long ring of DNA, but your prokaryotes also have what we call DNA plasmids. They are smaller rings of DNA, and they're what make bacteria capable of evolution very quickly. These plasmids can be traded with other bacteria like a flash drive so that they can get new sets of instructions from those plasmid DNA. So again, those are like portable DNA fragments that can code for new genes. So what protects the cell? What keeps the insides inside it, and what keeps the things outside? So for all cells, they have a plasma membrane that separates the inside and the outside. But for bacteria and plants, they have a special cell wall for an extra layer of rigid protection. So let's describe this in detail. Your plasma membrane is made up of what we call a phospholipid bilayer. What that means is that there are phospholipids, as seen here on the right. These are molecules that have a hydrophilic head, meaning they like water, and a hydrophobic tail, meaning they hate water. So because of this structure, the heads are the ones that interact with the water inside and outside the cell, while the hydrophobic tails hide away from the water in the environment. So what happens is that when you have a lot of phospholipids, it forms a sandwich where the hydrophilic heads are outside of the sandwich, they're like the bread, while the hydrophobic tails, yung palaman or the filling of the bread, inside the phospholipid bilayer. And because of this phospholipid bilayer, there are unique mechanisms for controlling the transport of different molecules inside and outside of the cell. So the plasma membrane is also described with the fluid mosaic model. This is the way that scientists think that plasma membranes are arranged. So it's fluid because the phospholipids form like a sea or an ocean, while proteins and carbohydrates and cholesterol float around in that sea of phospholipids. So because of that, there are unique characteristics and mechanisms that the plasma membrane can use for transporting different types of molecules. We're going to talk about that later in the quarter. On the other hand, for plants and bacteria, they have a cell wall, the extra layer of protection, and they're different between bacteria and for plants. So particularly for your bacteria, they have what we call your peptidoglycan part of the cell wall. So in your bacteria, in your gram-positive bacteria, they have thick peptidoglycan. Again, that's a polymer that is very special and helps protect the bacteria cell. So the peptidoglycan, because it's very thick, that is where the crystal violet gets embedded. On the other hand, for gram-negative bacteria, the crystal violet cannot get to the peptidoglycan layer because there's an outer membrane. And the peptidoglycan is very, very thin in your gram-negative bacteria. So again, peptidoglycan is a polymer that strengthens the cell wall of bacteria, and it's where the crystal violet in gram staining gets stuck. In your cell wall of the plant, instead of peptidoglycan, its main strengthening molecule is the cellulose. Cellulose is made up of carbohydrates that are put together in a special way so that it is very, very strong and rigid. In fact, our enzymes in our human digestive system can't even digest cellulose. That's why you have to chew your vegetables and fruits very, very well, because you can't digest the cell wall of plants chemically. You have to break them down mechanically by chewing very well. So next, what is the fluid inside the cell? What are the other structures floating in? We call that the cytoplasm. Well, more specifically, the cytoplasm refers to all the fluid and material inside the cell, but outside the nucleus. So what that means is that the cytoplasm includes both the cytosol, which is the fluid inside the cell, and the organelle, so any specialized structure inside the cell. Now, we mentioned that the organelle is a term that is quite complicated because organelle can mean many different things. For our study, for our lecture, it's any specialized structure. For other references, organelle can mean only membrane-bound specialized structures, meaning if there's no membrane to put it in a bag, it's not considered an organelle in some references. But for our discussion, when we say organelle, it's all of the specialized structures in the cell. So what gives the cells their shape and form? It's the cytoskeleton. So the cytoskeleton is made up of filaments that keep the structures and the cell all together. So it anchors everything. So let's hide my face. We have three main types of... We have three main filaments that make up the cytoskeleton. We have the microtubule. It's the largest type of filament, and it also is involved in the mitotic spindle and the flagella and the cilia. Your actin or your microfilament is the smallest kind of filament, and it functions in muscle contraction. So when you flex your muscles, the actin is actually working with another filament called myosin. But again, microfilaments are important for that contraction, as well as primitive movements, such as in the case of amoeba, the way they just crawl, like spread out and then just pull themselves along. That is due to the microfilaments. The intermediate filament is a middle-sized filament that keeps stuff in place. It anchors and scaffolds the organelles. So the mitochondria, everything else, they are stuck in the intermediate filaments, like a film. Next, what do cells have to keep stuff in? What can they have something to organize stuff? Can they have bags? So they do. They are called vesicles. So they're like membrane bags. The membrane acts as an outer covering. And there's actually a lot of types of vesicles. So we have small vacuoles in all types of cells that are literally just membrane bags for water, food, or waste. On the other hand, plant cells are have a very large central vacuole to store water and a few organic molecules just in case the rain doesn't come very often. So it also controls turgidity, or how rigid the plant cell is. The more water is in the central vacuole, the more rigid the plant cell becomes. If there's less water, it becomes kind of flaccid and flimsy. Next, we have in your eukaryotes the lysosome and the peroxisome. Your lysosome is something that breaks stuff down using hydrolytic enzymes. It uses hydrolysis, so it breaks stuff down using hydrogen-based molecules, and also makes water as a byproduct. So it's necessary for digestion and recycling. So it can digest food, it can digest broken organelles, it can digest entire cells, especially if your cell is a bacteria cell that's invading a host, a eukaryote host. On the other hand, we have peroxisomes, which actually break down stuff using oxidative enzymes. So it's just the same function, but different type of enzyme. But this oxidative enzyme has a bit of a flaw. It has a byproduct, which is hydrogen peroxide. So it breaks down things like fatty acids, which are parts of your fats. It also breaks down ethanol, the component of your drinking alcohol. So the problem is it makes toxic hydrogen peroxide as a byproduct. But it also can break down this hydrogen peroxide so that it keeps your body safe. It keeps the cell safe from its own byproduct. So your peroxisome is actually why when you put hydrogen peroxide on your wounds, it bubbles. That bubbling is because the hydrogen peroxide is being broken down into water and oxygen gas by your peroxisomes. Alright? So next, we'll be taking a break and we'll continue in the next video. See ya, guys.